Hello, and welcome to this video, where today we will be discussing the process and reactions of glycolysis. What is glycolysis? Glycolysis is a pathway that is involved in almost every organism. This pathway breaks down the molecule glucose using enzymes to produce energy for the body to function. Although other molecules are formed as end products, the main purpose of glycolysis is to form energy. This pathway takes place in the cytoplasm of the cell. Before we can talk about the glycolysis cycle, we must understand what glucose is. Glucose is a common simple sugar, part of the carbohydrate macromolecule family, that is found naturally in most foods. It has a very recognizable structure, 6 carbons, 12 hydrogens, and 6 oxygens, and is seen commonly throughout biology and chemistry. It is also the main sugar in blood, which can be explained by glycolysis. This is because the simple sugar travels throughout the body to different cells via blood vessels, allowing the cells to make energy. The preparation phase is a part of the glycolysis process that turns glucose into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which also turns into glyceraldehyde 3-phosphate. The reason this stage is called an investment stage is because this phase uses 2 independent ATP molecules as an investment to produce 4 AT molecules during the payoff phase. We will go into further detail later into the video when discussing the payoff phase. In the first step of glycolysis, there is a molecule of glucose that is added with ATP and catalyzed with hexokinase, which turns glucose into glucose 6-phosphate. Due to the phosphate groups on the ATP binding to glucose, which results in the ATP being converted into ADP. This step is irreversible. After it is converted to glucose 6-phosphate, it is then converted to fructose 6-phosphate by using the enzyme phosphohexose isomerase to open the structure up and convert it into a 5-membered ring. This step/reaction is reversible. Using the 6-phosphate, a phosphofructokinase 1 enzyme is used to catalyze the 6-phosphate to fructose 1,6-bisphosphate by using ATP. One of the ATP's phosphate groups is added to the structure, which causes it to be converted to ADP. This step is also irreversible. Using the fructose 1,6-bisphosphate with the enzyme adolase to catalyze it, it can be converted to 2 glyceraldehyde 3-phosphates and a dihydroxyacetone phosphate. The pathway continues on with the 2 glyceraldehyde 3-phosphates, detailed in the figure to the left, while the dihydroxyacetone, however, can be converted into glyceraldehyde 3-phosphate by using the catalyst triose phosphate isomerase, detailed in the figure to the right. This phase is reversible, along with the dihydroxyacetone to glyceraldehyde 3-phosphate reaction. In the payoff stage, it all starts to come to an end. The overall pathway starts to make a profit by turning the 2 ATP invested earlier in the preparation phase into 4 ATP molecules, which gives us a net profit of 2 ATP molecules. Using the 2 glyceraldehyde 3-phosphates, the 2-phosphate plus 2 NAD plus molecules are added to the reaction with the glyceraldehyde 3-phosphate dehydrogenase enzyme, which catalyzes it. The phosphate group is added to the glyceraldehyde 3-phosphate molecule, which causes it to remove the hydrogen off the inorganic phosphate. Then, using the NAD plus molecule, it is reduced to NADH. This phase is reversible. The oxidized phosphate is now bound to the glyceraldehyde 3-phosphate, creating 1,3-bisphosphoglycerate. Using the 1,3-bisphosphoglycerate with the ADP from the first step and the enzyme phosphoglycerate kinase, the ADP is converted into ATP from the donation of the phosphate group of the 1,3-bisphosphoglycerate, yielding ATP and 3-phosphoglycerate. This phase is reversible. Using the catalyst phosphoglyceromutase and 3-phosphoglycerase, the histidine side chain is activated and converted into 2-phosphoglycerate. This reaction is also reversible. 2-phosphoglycerate is converted to 2-phosphoenolpyruvate using the enolase catalyst. During this reaction, dehydration is taking place and water is lost and produced from the 2-phosphoglycerate and a double bond is formed. This phase is reversible. The final step of the glycolysis process is the catalyst reaction with phosphoenolpyruvate and ADP that turns it into pyruvate using the enzyme pyruvate kinase. It does this by reacting with the phosphate group from the phosphoenolpyruvate, which turns ADP into ATP and the phosphoenolpyruvate into pyruvate. This step is irreversible. In those detailed and complex 9 steps, this is the net reaction summary of it all. 1 glucose turns into 2 pyruvate, 2 NAD pluses are invested and reduced to create 2 NADHs, 2 ADPs plus 2 phosphate groups are invested, so 2 ATP are used to create 4 ATPs, netting a 2 ATP net gain. Thanks for watching, and I hope you learned a little bit more about glycolysis today.